I think the fact that they have an opportunity, Down syndrome, to do a lot to uncover the cause of this disease, give opportunity not just to help the folks that have Down syndrome who are getting older, but the general population. So what I'm going to tell you is a little bit about the as it impacts Down syndrome, but also with reference to the general population, and give you an update on what we think about and some of the research findings that we have, and I think in the end project a very hopeful message regarding the very strongly hoped for ability and as possible actually be able to say to families that we know how to prevent Alzheimer's disease in people with Down syndrome. We can't do that today, but eager to be able to say that, and we're hopeful that we will be able to say that in years to come. So we'll go to the next slide. I think this is the general hypothesis that people are probably very comfortable in, but let me just say it. So all of the changes that we see in people with Down syndrome, in their brains, their livers, their kidneys, their bone, all of these changes are due to an increase in the dose, i.e. the number of genes on chromosome 21. Down syndrome is a disorder in which there's an extra copy of chromosome 21. It's a perfectly normal chromosome with perfectly normal genes. It's just that there's one extra copy. That is the genesis of all the changes. It doesn't mean that one easily trace from a gene to a consequence. It does mean that directly or indirectly, that's the problem. So next slide. So, and what I have here in front of you is a slide that shows on the left-hand side the chromosome 21 for humans, and it's green. And so there's a little black dot separating a bigger green dot and then a long green column. The black dot is the middle of the chromosome. It's called the centromere. And to the north of that, if you will, upside is a very short segment, and then below is this very long column. That's a representation of all those genes that are present there. There may be as many as 500 genes on chromosome 21. It's the smallest chromosome. It still has a lot of genes on it. So that's the chromosome we're talking about, and I'll come back in a little bit to talk about the mouse models that are modeled really genetically after human chromosome 21. So slide. What do we think, and what do we study? Well, our hypothesis is that the gene for the amyloid precursor protein, so-called APP, that the APP gene plays an important role in brain structure and function. That's really well known, quite frankly. Increased APP gene number, or dose, if you will, is responsible for Alzheimer's disease and Down syndrome, is increasingly accepted as the case. Study how it acts, and we think it acts in part by the ability of brains to function normally, to send information from one part of the cell to the other, and that ultimately results in neuronal dysfunction of those cells that transmit information in the brain and to their death. And with the very consequence of a dramatic decrease in cognition over baseline and death. Next slide. Etiology of Alzheimer's disease and Down syndrome. What you see on the left is two brains, each in half. On the left-hand side of that panel, the healthy brain. Notice that it's quite full. There's a rich assortment of tissues there. The surface is the gray matter, and the material inside is the white matter. The gray contains the neurons. The white matter contains their axons. Notice on the right half of that panel, the picture of the brain of someone with advanced Alzheimer's disease, or with someone who's elderly and has advanced Alzheimer's disease and Down syndrome. Notice that the surface staining, the gray, if you will, the brown in this picture is thinner. There's white matter as well, And the spaces in the brain that one can barely see on the left-hand side are much enlarged. And notice, well, toward the bottom, there's a fairly, and I wish I could show you this on my pointer, but toward the bottom of that slide, that half, notice the space. It's kind of an oblong space. That's called ventricle, that is near the hippocampus, and it's big, but the hippocampus shrunken. And on the right side are the very characteristic neuropathological features of Alzheimer's disease. In the middle are so-called neuritic plaque. The plaque is composed really of a number of fibers that converge on a collection of amyloid protein, and that amyloid protein is produced as a result of the processing of ADP, the gene excess that we think is so important in Alzheimer's disease and Down syndrome. On the side, as it's called neurofibrillary tangle, it's really a congealed mass of protein you've seen in the cell bodies of neurons and in their processes. The bottom of the states the case. The pathology of Alzheimer's disease, what I've just talked about, emerges in everyone with Down syndrome by age 40, and dementia ensues on average in 50%, that is, by age 55. So slide. Position for Down syndrome and Alzheimer's disease. If there are fundamental links between pathogenesis, that is, disease causation in Alzheimer's and Down syndrome, and can define the links and mechanisms in syndrome, then one could integrate work in Alzheimer's disease and Down syndrome to diagnose the earliest onset of Alzheimer's disease pathogenesis and build more robust ability to develop, test, and validate treatments. So let's do these one by one. The first is, are there fundamental links? Next slide. Are there links between pathogenesis and in Alzheimer's disease and Down syndrome? And the next slide speaks to this. So the pathology is very similar, if not identical. It's absolutely the case if you give a neuropathologist a slice of a postmortem brain sample from someone with Alzheimer's disease from the general population and from someone with Alzheimer's disease from Down syndrome, they can tell the difference. Very characteristic pathology that's not so emphasized by many people, but we think it's very important. It's something called endosomal pathology. So let me go to the next slide and tell you a little bit about these endosomes. What you see is a kind of a cartoon of a cell. And the white space at the top is the outside of the cell. And it's that there's material that is taken, little cargoes are taken into these little pits, internalized. And those internalized vesicles, they're kind of light yellow color. Those internalized vesicles carry terribly important information to the cell and its nucleus from the outside of the cell. The population of those endosomes are called early endosomes. Quite frankly, those early endosomes, as well as other endosomes in the brains of people with Down syndrome, are quite abnormal and quite early. Next slide will show you some of this pathology. Notice staining these endosomes in brains of people with Down syndrome at different ages. Now, at the very bottom, you see there's an adult control, and there's very speckly staining there. But look at someone with adult with Down syndrome. There, those are quite large. Look at the arrows pointing out those early, very markedly enlarged early endosomes. And note well in the top panel that there's evidence of this pathology, even prenatal period, 20 weeks, 28-week fetus. So this enlargement is both sporadic AD, that is in the general population, and in Down syndrome, quite way before those classical markers that I mentioned before, the atrophy of the brain, the plaques and the tangles. Next slide. Another point is that pathogenesis, that is the disease process, proceeds over decades. Next slide. Let me just teach you a little bit about this. It's very interesting, and this is a one from Paul Azin and his colleagues here at UCSD. Note that we can, on the left hand, can go from normal to abnormal in that arrow, and on the bottom, we go from people who are pre-symptomatic to those that have early problems with cognition, to late problems with cognition, but that are minimal still, to dementia. And notice that the lines then mark out, if you will, different changes that occur over time. If you will, this kind of bright purple line, that's the volume of the hippocampus. Notice that over time it becomes more and more abnormal. Notice that it in fact becomes abnormal way before people have dementia, as reflected in the purple line at the bottom. And all other markers are also changing over time. So for the green line for amyloid imaging, the light green line, that becomes abnormal quite before, long before there are changes in cognition. Now, next slide, if you map out these changes over time, let's take just two of them. So this in the red line is the prevalence of dementia in people in general population. This is AD dementia in the general population. Note that if you're 60, the chance of having Alzheimer's disease. If you're 90 or 100, the chances are much higher. Next line will show you, it's a very nice little line, it's a green line. It shows you when those plaques, those neuritic plaques, become evident. Now, the line is moved over to the left. Now, if more push of the button, and we'll show you in the blue dots when, in fact, brain imaging studies show the amyloid is present. The next little line says that there's 15 years, pushing again, there's 15 years between what one can see as the presence of plaques in the brain, the characteristic marker, and onset of dementia. Next, what's the case for Down syndrome? Well, really different. Pathology is present between ages 35 and 40. We know that half of the people develop at age 55, and that greater than 90% have dementia at age 70. So in the difference in time between when pathology is present and when dementia is present, it's about 15 years, whether you're someone with Down syndrome or whether you're someone in the general population. It should be very clear to people from this exposition that Alzheimer's disease occurs routinely in people with Down syndrome, and the process, while it might have some differences, persists over very similar time course. Bottom here is if you live long enough with Down syndrome, you're very highly likely to develop not just the pathology of Alzheimer's disease, but also dementia. Next slide.
And Bill, I just want to take a second because we've had so many people join us to remind people to send questions to me, Ellen Oliver, via chat or Q&A, and we will answer them all at the end of the presentation.
So what about the cause of Alzheimer's disease syndrome? And so pathogenesis, again, implicates this gene for APP. So these points. The genetics of Alzheimer's disease points to APP alone. A copy of APP alone is able to cause Alzheimer's disease. So you need an extra copy of APP to cause Alzheimer's disease. Most importantly, if you don't have an extra copy of APP, it appears that you do not have Alzheimer's disease, even though you have Down syndrome. So let's look at the next slide. This is work from two different groups. A paper published a long time ago. There was a 78-year-old woman who had syndrome. She had very many of the characteristic features of Down syndrome, but there was no evidence of dementia on clinical exam or by MRI scan or at postmortem examination, and they looked very carefully at her genome. And she actually a partial trisomy 21. She had only 2 copies of APP. Colleagues more recently have been following a 77-year-old man with Down syndrome. Followed him for 7 years. They have seen dementia in this man. Now he's way beyond the point of having dementia and Down syndrome. They measured A beta 42 in serum. It's not increased. That imaging really is not abnormal, and he is also known to have only 2 copies of APP. So I think we can say that an extra copy of the gene for APP is causative of Alzheimer's disease in Down syndrome. Next slide. So let's go to our hypothesis. It really focuses on the APP gene dose, and it really asks the question, how does it cause dysfunction and death of neurons? Next slide. So we have tried as best we can in mouse models to define how that all works. How does it happen that a copy of APP causes Alzheimer's disease and Down syndrome? Next slide. So back to our slide of genes, but now we're going to look at those names and at those genes that are so important. So human chromosome 21, again on the far left side, all of the human 21 are found, not all of the genes, but the regions on human 21 are found on mouse chromosomes 16, so MMU 16, 17, MMU 17, and MMU 10. One can, by knowing which genes are present in both the human and mouse models that have one or more of these segments. Most of the work has been done so far in the TS65DN mouse. There's a little yellow box around that mouse. It's not a perfect mouse, but it has many of the genes in APP that are present on human chromosome 21. Next slide. So we know when increased APP gene dose in this mouse results in increased levels of the protein and its products. We know, like as is the case in Down syndrome and Alzheimer's disease, endosomes are enlarged. These mice show age-related dysfunction and degeneration of neurons in regions that are also affected in Down syndrome and Alzheimer's disease. In all cases studied so far, we've shown the extra copy of the APP gene necessary for degeneration recently shown, in fact, endosome dysfunction. These enlarged endosomes are almost certainly contributing in an important way to the pathology that we see. Next slide. This is a paper from Randy Nixon and his colleagues. In the left-hand panel, those arrows are pointing to endosomes. In the lower right-hand panel, what happens when you knock that extra copy of APP out is that staining markedly decreases. Next slide. Of brains that are older in these TS65DN mice, that cholinergic neurons that die have terminals in hippocampus that are filled full of APP, and that APP is in these enlarged early endosomes. And next, about endosomes. We care about them a lot because the endosome carries the signal to neurons talking with one another, trophic factors. And so what you see is I have a grayish blob on the right and a blue neuron. Its body, its axon on that tube, and then the ending is the terminal. And what's the inside of there are these little circles, and the little circles are a cartoon that represents the little package, if you will, these endosomes that carry information from one neuron, in this case the gray neuron, to the neuron to keep it well and healthy. And it's to tell it, stay alive, still here, we need to still connect with one another. If one compromises this process, one is very likely to compromise the viability of the neurons. And in fact, next slide shows you, if you will, that when injected NGF, this trophic factor that's so important for these cholinergic neurons, when we injected in hippocampus, we saw that much less was transported back to the forebrain, to these neurons that require it in the TS65DN mouse. Next slide shows an overview of all those genes that we might have thought was important. Slide. That should be important. The only one that we've identified so far is the gene for APP. If you print the extra copy of APP from being expressed in a mouse, and say it this way, we use genetic tools to eliminate one extra copy of APP. When we did that, these neurons that did not degenerate, and there was another group of neurons in the brainstem, very characteristically degenerated in Alzheimer's disease and Down syndrome, also do in this mouse model. Taking out that extra copy of APP prevented the degeneration. is our target, and how do we proceed? And what is about APP gene dose that matters? Next slide. Well, is that we can look at the individual pieces of APP and figure out what's going on. So in the very middle of the slide, that green column, that green stick, is a cartoon of the protein APP. If to the left, you see that it's split into various pieces. These various pieces are come off in a slightly different way if you look to the right side. Called A beta or AB, that small little cylinder on the far right, it's toxic. C99, it's toxic. And the whole full-length protein right in the middle, it's toxic. All of those molecules create problems with endosomes that create problems with trafficking of trophic messages. They're all implicated in the disease. Next slide. In APP gene dose, with products, the ones I just mentioned, cognition of protein in an enzyme called RAB5, which increase in endosome size, and that really compromises transport of these signals, with thereby becoming sick and then dying. Next slide. So, can we integrate work in Alzheimer's disease and in Down syndrome to study pathogenesis and diagnose onset? And of course, here's a great thing to try to imagine now. We have a population of folks that we know will get the pathology of Alzheimer's disease. We can study these folks and to characterize when it is that these changes start to occur. And importantly, not just when they start to occur, but when they begin to have problems that impact cognition and function. Next slide. Now, what fits to the general population? Well, here's a cohort of individuals markedly at risk for Alzheimer's disease. They're identified just before the onset of dementia. It's a relatively healthy group. We can examine changes longitudinally over time, and we can construct efficient prevention trials. If we get the right medicines, we can actually prevent Alzheimer's disease in this population. The first population to benefit from the new therapies for Alzheimer's disease may well be people with Down syndrome. What a great thing for LuMind if LuMind makes possible prevention of Alzheimer's disease in people with Down syndrome and allows this work to teach the rest of the world how to prevent Alzheimer's disease in other folks. Next slide. And robust ability to develop, test, and validate treatments. So let me go to the next slide. We've got a number of ideas along these lines. Simple. If we're going to get APP, we could try to reduce the level of the messenger RNA for APP. And we've gotten data from a collaboration with ISIS, a company here near San Diego, that a small little nucleotide that binds the APP mRNA reduces the level of that mRNA. It enhances its degradation. But how cool to think about a therapy for preventing Alzheimer's disease and Down syndrome that targets the messenger RNA for APP. Second approach is to immunize people, reduce the levels of this peptide. Remember that little cylinder on the far right-hand side that was toxic. We've done animal studies that show that it works, and a trial sponsored by AC Immune and NIH will soon begin in San Diego in adults with Down syndrome. Attempting to understand, can we deliver that vaccine safely in people with Down syndrome, and if so, can we actually use immunization against beta to, in fact, protect against Alzheimer's disease. And then finally, we've here at UCSD with Steve Wagner's help, his leadership, a novel class of small molecules I think is going to make a huge difference by enhancing the degradation of this beta peptide. And let me just show you some data for that. This is work done in the mouse model of Down syndrome. And what I'm showing you is the amount of these proteins. This is this beta-42 peptide. Notice that we did both 2N, that's the normal mouse control, and the DS mouse model. either vehicle, V for vehicle, or with this thing called a gamma secretase modulator. Essentially, this molecule, this small molecule, enhances the efficiency of processing of ATP so that you get less of these toxic small peptides. Notice that the levels of that peptide, that A-beta-42, is significantly lower in the brain of both control animals and the animals that model Alzheimer's disease or Down syndrome. Notice, in fact, that in the TSM cortex, the level of that molecule is now lower than in the wild-type control not treated with TSM. Next slide, the same was true in plasma, where changes in plasma are even more dramatic. And the next slide shows you that we're having an effect on those C83 and C99 molecules, those transmembrane stubs that I spoke about very briefly. Even the levels of those are going down with GSM. Now, we're encouraged then that there are strong links in pathogenesis, that we those links and discover their mechanisms, one of which is the effect of APP gene dose on endosomal function, that we work in Alzheimer's disease and Down syndrome, and we're so eager to push this forward, and so increasingly we're getting inquiries from industry and as how might they now invest in studies, clinical trials in Down syndrome. And finally, I really believe that targeting APP is quite rational, and just mentioned the early results are very encouraging. Next slide. So to support us over time, we appreciate very much all the terrific support we've received, and the next slide will tell you about the people that have done such a great job, especially thankful to Qingbiao Wu, Steve Wagner, Abe Weissmiller, Pern Zhu, and Artie Rebriglia, and colleagues at AC Immune. And Ellen, I'm through. That's all I have.
I have unmuted Jamie, and Jamie, you've got controls, and welcome Dr. Jamie Edgin, everyone.
To be here. Today I'm going to be talking with you about one line of our work at the University of Arizona. We have 2 lines. One of them is examining assessments, and we worked on assessments such as the Arizona Cognitive Test Battery. We're now developing an assessment for younger children. I'm not going to discuss that so much today because we have transferred it and we're in the process of transferring it to an iPad. So we're still in piloting work on that assessment. Today I'm going to discuss with you our work on sleep and obstructive sleep apnea and sleep disruption in individuals with Down syndrome in relation with cognitive outcomes. Let me control this. Okay, there we go. It's an area that's quite a concern for parents of individuals with Down syndrome, and sleep is a medical comorbidity that might affect a number of aspects of cognitive function in individuals with Down syndrome. So it's a lifespan issue starting from infancy, as I'll show you in some of my slides today, extending even into the adult years and the years of dementia that we've been discussing in the talk. It's been a mystery to people exactly what the functions of sleep are, and we think of it as a cognitive state or a state that might be important to cognition. Or you know more about what sleep is influencing, you know that's a state in which there are important sort of brain functions happening, sort of integrating across neural systems that will and possibly support adequate brain development as well as learning. And so from the last 5 years, there is evidence to suggest that sleep can actually be a state that facilitates learning and even facilitates possible language and the acquisition of language concepts. So it's a very important state, and it's a very concern for individuals with Down syndrome and their families. Sort of looked at sleep as being such an important state for learning that those have written about sleep, including William Dement, who's at Stanford University, chapters in their book called Sleep School, in which they sort of emphasize that this can be a very important state for learning. I just wanted to go go through the different brain states that relate to different stages of learning, because it might be new information for some of you who are listening. So on the left there, you see the different stages when someone goes from an awake state to a very deep stage of sleep. So you can see that that weekly line there is an EEG measurement. And so basically, you can see that that's an awake state with a very sort of high frequency EEG measurement that indicates the person awake. And then as they move into the deeper stages of sleep, it's more likely that they might engage in sort of larger oscillations between different brain regions that will allow for the development of those regions and also sort of the acquisition of knowledge, plasticity, enhancing of representations that have been during the day. Moving to stage 3 there, or what we call slow sleep, which is now stage 3. We know that from findings in the typical cognitive neuroscience literature that if you stimulate the brain at certain peak points of that wave, you can actually enhance learning and the retention of knowledge. So there are some deep stages of sleep that individuals go into that are important for learning and for retaining information. And unfortunately, if someone has a sleep disorder, they might not enter into these deeper stages of sleep that would allow for them to retain knowledge. So sleep disorder that often can keep individuals from consistently staying in deep stages of sleep or causes them to transition between deeper and deeper stages of sleep is obstructive sleep apnea. We know that individuals with Down syndrome are at quite risk for this disorder. Apnea is Greek for without breath, and it's caused by the clutter of the airway. So you can see here this depiction of that when one is sleeping and there enters through the mouth and the nose. And unfortunately, all of us have been designed with this sort of narrow airway. And some of us are more likely to have this airway closed during the night because of different factors such as obesity, low muscle tone. In Down syndrome, they're particularly at risk because of the craniofacial structure and the airway being naturally smaller. And so when this airway does close and during the night, you see this oxygen reduction, and the body has to basically arouse itself to get out of that state, which is the oxygen is reducing so much that we need to actually increase the oxygen level. It's a bad state to be in. So this is a process that someone goes through when they're having an apnea episode. So the person falls asleep, the upper airway relaxes, there's snoring, so there's resistance in their airway. That's what the snoring is. The throat closes or it obstructs, and you can see that the breathing will stop. There's an oxygen reduction. The person actually has an effort to breathe. If you can see someone who's in this state, it looks like they're breathing. They're just not getting any air through airway. And then you have that oxygen level fall. The brain senses that this oxygen has been reduced, and it makes the person partially wake up. They may not wake up fully, but they may transition between different EEG states or the deepest stages of sleep that would keep them from staying in those deeper stages of sleep, or possibly even getting into the REM stages that are in the later part of the night. And then what will happen is the body arouses, it moves us between different sleep stages, and then the breathing starts again, often with a gasp and a body movement. Okay, people can cycle through these different process many times a night. So this can be, for those who are most severe, this can happen 60 to 100 times an hour. So in Down syndrome, there's an increased risk of having obstructive sleep apnea. So the lowest rates that are usually reported are 30%. Most of the studies that I see report about 60 to 80%, and that's fairly consistent across the lifespan that we see individuals with Down syndrome have an increased rate of obstructive sleep apnea. And they also have other sleep problems, so it's not just obstructive sleep apnea, there are central apneas. These are higher than other developmental disabilities, including autism and those who are born extremely premature. So there are evidence that suggests that individuals with Down syndrome have more fragmented sleep than other developmental disabilities groups, a particular issue for them. A study recently published this came out last week, was looking, and this is a group from Annapolis, including Randall Roper, showed that a group of clinic-recruited individuals with Down syndrome who were aged less than 6 months, so these are infants, 177 of them. The parents were asked whether or not they were concerned about their sleep, whether they were showing signs of any sleep disruption. And what they found was about a third of the sample reported they had some concerns about that, and then they were screened with a NAP polysomnography study, which is it's a way of telling whether or not they have obstructive sleep apnea. And they found that in that 33% that were screened, that the large majority of them actually had moderate to severe obstructive sleep apnea. So there were only, I think, 3 children who didn't show that apnea in that group, and most of them were having at least 6 events or more per hour. So when parents were reporting that there were issues, they actually were correct in seeing this apnea in the children. There was a large percent of them, 2/3 of them, actually didn't show any concerns. But as I'm going to show you in the rest of my slides, it's uncommon for parents not to be able to pick up the apnea events. And so one of the things that we're trying to work on is more screening and more awareness of apnea in this group. So the first study that we published looking at the relation between obstructive sleep apnea and cognition was last year. And we recruited a community sample. These were not individuals that were particularly worried about obstructive sleep apnea, but we tested 31 children with Down syndrome, ages 7 to 13 years. We had a group who did not have apnea, and we also had a group that had apnea. So they had 1.5 or more events per hour. So that was the clinical cutoff we used. So we compared them on a lot of different background factors, and they were equivalent on a lot of different variables, such as MI, et cetera. And we looked at the relation between their cognitive function and their level of apnea, so whether or not they had apnea or not. We used a gold standard polysomnography assessment, which basically gives us an idea of how many apnea events they have per hour. It lets us know what sleep stages they're going into, so whether or not they're entering in these deepest stages of sleep, as I discussed before. Tells us how many times they're arousing between different EEG states, and also let us know what level of oxygen desaturation they had. So we have in-home polysomnography for this study. We related their apnea scores to a neurocognitive assessment that was conducted within 3 months of that study. So that was the Arizona Cognitive Test Battery for Down syndrome. And what we found when we looked at those who had apnea versus those did not was they actually spent more time in the lightest stages of sleep. So you can see there on the left, that's stage 1. They're spending more time in those lightest stages of sleep that had obstructive sleep apnea. And they spent less time in this slow wave sleep, or that deepest stages of sleep that I've told you about, that we know from the cognitive. So neuroscience literature can actually be quite important for the consolidation of knowledge. Okay, so that's what they look like when we looked at their different sleep stages. Their cognitive outcomes, so comparing the Down syndrome with and without apnea, who were matched on a lot of other background factors, age, gender, et cetera. And we could see that kids that had obstructive sleep apnea actually had a 9-point lower IQ than the kids who did not have apnea. And we found that they're more likely to have difficulties with flexible cognition. So you can see there on the right, that's the difference that we saw, that the group that had apnea actually looked like they had more difficulty sort of switching between rules, doing something new after they had learned an initial rule. Both of these areas are ones that we see in populations with in the typically developing population if there is a sleep issue or if there's apnea involved. So this is a big that is relating in Down syndrome as well to having sleep difficulties. Given these findings, we began to be quite concerned because of reports such as the one I presented to you from Indianapolis about obstructive sleep apnea and sleep disruption in very young children, and how this might occur in very important stages, very critical periods for cognitive development. And so went to younger children. So this is a group that we've studied now that's 2 to 5 years. And we measured their sleep using activity, which is a movement monitoring system that helps us to know how fragmented their sleep is, how many times per night they're arousing, how many times they're moving. We related that measurement to parent-reported language development. So it's the MacArthur CDI, which is a language measure that has been often used in Down syndrome. It's one of the most studied language measures in young children with Down syndrome. We also measured natural language samples, so we had monitors that picked up about 6 hours of their language. And then given that sleep disruption has been related to exacerbated autism symptoms in those who have autism and those risk for autism, we actually looked at behavioral ratings of autism using an autism screener. This is any data that we get from these measures. We found about 66% of that sample of toddlers with Down syndrome had poor sleep that was very different from the typically developing controls we saw. So over the course of the night, you can see those blue periods are when they're sleeping. And all the black lines within that blue period are points in time in which the toddlers were moving and showing movement. So 66% sample showed poor sleep. We then related that to different cognitive and behavioral outcomes. And so this study, as you can see there in the lower right-hand corner, is actually an invited paper for revision. So I have to declare that these findings are preliminary. They're not published yet. We're in the process of publishing them. It's likely we will, but they are very preliminary. And I think that some of these findings are sufficient to suggest that we should continue and do more work in this area, and we have some grants into NIH to try to expand on these studies based on these initial findings. So this first slide I have here is the relation between the sleep problems and autism symptoms. I know this is a screener for autism. To diagnose autism, you actually have to use a gold standard battery to do that. This screen has a lot of false positives. But when we looked across these different groups, you can see there the typically developing controls there on the right. They're about 5.2%, which is a little elevated. Good sleepers were not significantly different. And then there is this little bump in the poor sleepers. None of this is statistically significant. So we don't say anything about it other than something that we want to take into account when we're looking at other findings in this group in relation to sleep. It's also something that leads us to want to do more research. The language differences in those who had poor sleep on the left, the good sleep, and then the typically developing controls. And we did see differences in language function. So that was our strongest finding and what generated the interest in this paper that hopefully we will be able to rise and get into the literature. So we did see that those who had poor sleep had sleep efficiencies that low and consistently low across those 5 nights. Actually, about 190 words difference in their parents' ratings of their vocabulary scores. That those kids who had poor sleep combining words about 30% of the time. The good sleepers were combining words about 80% of the time. And you can see that on the right, on that lower graph, typically developing controls were about 100% of the time. So we're seeing they were combining words less. And then we also had findings that were objective reports from our language samples. So we had line coders go to language samples. They didn't know the sleep status of the children. And they coded the complexity of language they saw, or how sort of the length of the utterance that they heard in those samples. And we saw differences in the length of the utterance based on our coding. And controlled for a lot of different factors in these: age, controlled for the difference in the autism symptoms, and we still got these effects. What I do now is that in an older sample, there's a difference in cognitive function. In this top sample, we're seeing some findings that lead us to engage in more research, and it seemed to be centered on the language differences. But in both of these studies and in other studies by other laboratories, including Sally Schatz's laboratory, parent reports were not relating to sleep, not showing us these differences. And this suggests that we really do need more screening. So here you see the data from Breslin et al., this paper published with polysomnography. And parents, when they were asked if they snore or the child stops breathing or the child snorts and gasps, That's not relating to the polysomnography measures, and this has been found in other labs. So it does suggest that we need more screening approaches to pick up these signs in individuals with Down syndrome, and more children need to get gold standard sleep assessments. So back to the finding, the recent finding from the Indianapolis group. That group of the 2/3 of the parents that weren't screened, what was going on with those kids, and how can we get kids into more screening and really understand how big of an issue this is, and expand on our findings to really understand overdevelopment, what the impact of sleep disruption is going to be. So the preliminary results have led us to gain more funding from the Bill and Melinda Gates Foundation to conduct studies to follow up children from 6 to 24 months, children with Down syndrome, and to see whether or not their sleep disruption will relate to outcomes like how well they're combining words, not they transition to an autism diagnosis, and we're comparing kids with Down syndrome to a group of children who were born very premature, less than 33 weeks, and also children who are at risk to develop autism because of having a sibling with autism. So we're seeing them every 6 months from 6 months until age 2. And conducting a lot of different measures of their parent history. We're also giving them sleep assessment, relating that to gold standard sleep assessment to see if there's any signs that we can pick out that could be clues to parents that their child might have some sleep disruption. We know right now from our data, and I don't have a slide for this, but we know right now that kids with Down syndrome are actually napping for longer than their peers. So they're not transitioning out of naps as early as their peers, and they tend to nap for longer. So that's one sign that we're seeing in this early childhood period. Hoping this study will help us to get more signs as well. And then we're going to take that to their developmental milestones and their outcomes. We study, this is a PhD study of my graduate student, Gofferdina Spano, and this is done in collaboration with Rebecca Gomez, who is an expert on sleep and learning at the University of Arizona. And we're actually teaching kids words prior to a nap period and then seeing whether or not they can retain these words across that period. And mechanisms might be different for them that could tell us why they might not be learning across the sleep period, what sort of might be happening with their brain function across this period. So if you use to target a treatment to sort of change a particular brain state to help support learning across the sleep period. Summary, from the typical literature, we know that sleep is a key for health and well-being and learning. We're seeing differences in range function relating to sleep difficulties, Down syndrome. And given the amount of these correlations that we're finding, we're trying to support the use of sleep as an outcome variable in clinical trials. Parents have a lot of concern about this area. It would be wonderful to know how different therapies affect sleep, whether or not there might be sort of a breadth of therapies that could be used to influence this very important outcome. We know more treatment options that are needed. Early detection is also needed for obstructive sleep apnea and sleep disruption. Better tools are required. So we're starting a collaboration with an engineer who's actually looking at a system that includes an inner system, which is basically has an infrared beam of light that helps us to understand how the brain is functioning during sleep. He has the system set up. It's a very small package that actually can deliver this beam of light. And we're working with him. I've convinced him that he needs to do this in Down syndrome. And so we're working with him to see whether or not we can use that alongside some of the studies that we're running right now. So I'd like you guys to go sleep on what I've told you and wish luck, because we have some grants that are right now that into the NIH that are going to be reviewed very soon on these topics and to expand these studies, and hoping that they're successful. So wish luck. I just want to thank all of my collaborators. There are many people at the University of Arizona that have come on board to work with us on these different studies. So collaborating now with a group in Mexico to look at sleeping children in Mexico as well. And then Bob, who's at Banner in Phoenix, we're doing a study looking at aging in sleep in that study. So thanks to all of the funders of this research, and thank you for your attention today.
Thank you, Amy. I have you remain unmuted, and Bill, you are also unmuted because we do have a bunch of questions. The first one is for you, Dr. Mobley. In a person with typical trisomy 21, how many copies of APP are there?
A person that has typical Down syndrome has 3 copies of APP.
Okay. And related to that, a few questions on how close we are to bringing a therapy to market as it relates to APP.
So it's hard to know exactly how that's going to work out. What you can say is that there's a phase 1 trial that'll start very soon here at UCSD looking at this vaccination approach. We're that trial to be we're to be able to be known. Then probably talking 3 or 4 years before that vaccine would be available. The GSM approach that I mentioned, we'll file an IND using an existing GSM before the end of this year. That's 1 trials could begin as soon as next year, and with any luck, and with not many luck, with more than a little bit of luck, obviously, because it's difficult to make drugs work. But, you know, again, we're talking probably 3 or 4 years before we've got a therapy that might be tried, that could be tried in people with Down syndrome. So these are all guesses, and they're somewhat optimistic guesses, but want to be optimistic about this because we think what we're doing now is going to provide value, and so far there's just no evidence in animals that there's any toxicity for these therapies that we're looking at.
Okay, Jamie, I have one for you. Someone who wants to know about research showing data for children after intervention, such as a tonsillectomy, and with and cognitive improvement, and these children experiences increase in these areas after intervention.
So there, a couple groups have done that, and so one study, unfortunately the numbers have not been very high. They're very difficult work to do. And the study came out last year on Brooks. It was published by Brooks and colleagues. They saw a couple kids pre and post treatment on CPAP and increases in intention. I think they were older children in that case. There haven't been that many studies done in younger children. And what we're saying is that we need to period down in which we're actually treating kids from the sleep disruption. So a lot of the studies have been conducted actually in older children, and we need to get more detection early, and there may be differences in outcomes in that early period. That's when which is developing and so I haven't seen anything in the early childhood period in terms of having tonsillectomy and pre and post outcomes with cognition, just in the later childhood period.
Thank you. Another wants to know about proposals about the inhibition of DYRK1A that may be beneficial. I want to know your opinion on this. Bill, do you want to chime in?
Sure. I mean, this is a really important gene that's also in chromosome 21. Very likely that it's interfering with a number of different pathways in neuron function. And I think the studies to exactly how it might be working are yet to come. There was EDG is an inhibitor of this enzyme, and it was being tested in Europe. I haven't seen the data from the trial yet to be confident that I know what it shows. The issue with that small molecule is that it does more than inhibit DYRK1A, and so it's going to have to hope that one can test molecules that are even more specific for DYRK1A than ECG, but having that, I think a line of investigation that says that DYRK1A is important. Let's see if we can modulate its activity is a very important one. One that I think is more specific and possibly very useful would be the approach we're using, mentioned, using antisense oligonucleotides, ASOs, for APP. There one would be selectively targeting the messenger RNA for DYRK1A and conceivably might have even a more specific effect on that, on the level of the enzyme activity. But this work for the future, I think for the moment, terribly important research area and question, more to learn.
There's a bunch more questions, and I just want to let people know that I will be sending these questions to Bill and Jamie, and we'll be happy to answer as many as we can. So we probably have time for just one more, and I think I'm going to send this one to Jamie. Sleep obviously contributes to cognition, and they just had some questions about the end goal of your research, and are you finding ways and methods to get people with the needed sleep? And I also just want to, before we wrap up, let Edgin and Dr. Mobley know there have been many comments thanking you for your work. So go ahead, Jamie.
Oh, pleasure. So the goal is to support sleep in kids with Down syndrome and to find the ways to do that. We've worked a lot on sort of awareness and the initial correlations between sleep disruption and cognition because we think it's going to move students earlier on, more awareness that need to be good. So one that we are trying to support that is to get a better understanding of the mechanism that might keep the kids from learning. Okay, so yesterday I told you about my graduate student, Dr. D'Agostino. She's looking at learning across that nap period and to try to see whether or not there are particular phases of sleep that might be particularly disrupted. And doing so, that might give us some sort of about how we may intervene in novel ways. I can tell you that's kind of where we're going with this. There are a lot of other treatments that have been proposed, and what we're just trying to understand the mechanism that might be keeping them from learning. And we think that that's a good approach for us right now to understand that mechanistically. I can't have much more to the question. Getting to what we're trying to do.
Right, and we are trying to wrap up. So if you can give us a quick sentence on that, I don't know if that's too much to ask.
You know how early this is happening, how big of an impact it is, sort of, a wind of change in trajectory we can expect based on the app and the system itself, and to see if that's going to support earlier treatment approaches. But then mechanistically, we need to know exactly what might be happening in the sleep. Is it oxygen? Is it that they're not getting into deep stages of sleep? We need it mechanistically in order to help support the treatment of the sleep-cognition relationship.
Thank you, Dr. Mobley and Dr. Edgin and everyone for joining us today. From a quick look at the registration list, I know many of you are already involved in our organization, and we thank you for doing so. We provide these webinars as a way to say thank you, and so you can understand the progress that your donations fuel. We hope that after the webinar you feel as hopeful about the future for some of these discoveries and research as we do here at LuMind Foundation. For those of you who are new and seeking additional information about research, we invite you to be part of our community. The slide is showing the contact information for myself, Carolyn Cronin, our Executive Director, and Dr. Michael Harpold, our Chief Scientific Officer. It is also a short list of ways you can get involved. We want to hear from you, so please reach out. We invite your questions, ideas, and your interests. And thank you for joining us today. The webinar recording should be available in 24 hours, and hopefully you'll receive an email letting you know when it's ready, but we will also be posting it on our website and social sites, and probably Dr. Mobley and Dr. Edgin's Facebook groups are doing the same. We invite you to check those out. And again, thank you so much to everyone, and mostly to Dr. Mobley and Dr. Edgin for your presentations and for answering all the questions today.
Thank you.
So, okay. everyone. Thank you.